Hello, hi. Shall we begin? So today we are going to see the asexual reproduction in Oedogonium. Okay. Oedogonium we have already discussed its salient features or the general characteristics, its ecological habitat and things like that. So we have seen things like its general structure. It is having three kinds of cell. One is an apical cell, then second is a basic cell, and in between we have intercalary cells. Okay, and we have discussed that the reproductory cells are cells which are bearing cap-like structure. So if there is one cap, it indicates that it has undergone one division. If it has two caps, it indicates that it has undergone division twice. Okay, so we are going to see how this asexual reproduction happens in Oedogonium in this lecture. So what we are going to see is how this particular cell, this particular cap cell is going to be converted into a zoosporangium. Okay, we are going to see that thing. So this is the asexual reproduction cycle. Very simple. So you can see how it can be easily drawn as a pencil drawing in the exam. So with a practice, you can easily draw this figure with high speed. Okay. So these are uniseriate cells. We have already discussed that. That means these are arranged in a single line, end to end connected. So one of the cells during the favorable condition means when nutrients or water are available in plenty. That is a favorable condition for Oedogonium to reproduce asexually, as in volvos we have seen. So during the favorable conditions, one of the cells will undergo transformation and develop itself into a structure called the zoosporangium. So just imagine that one of the cells, one of the cap cells, not ordinary cell, one of the cap cells are cells which bear cap in a filament. So that cell is going to act as zoosporangium. So how it is going to act as a zoosporangium? What it is going to do for that? You can see the protoplast within the cell. That protoplast will shrink. So that it gets detached from the cell wall of that particular cell. Then the entire protoplast, which is having just one nucleus, it is going to be transformed into a zoospore. Okay, into a zoospore. The spore that is released from the zoosporangium is called zoospore. So when the particular zoospore is completely mature, or its development is complete, there will be a transverse split opening of, there will be a development of slit at the top of the cell where the cell and the cap joins. There will be a transverse split, and this split gets opened and the developed zoospore will be released, and it is coated in a vesicle. Okay, from the interior of the cell a vesicle is formed, and that vesicle will be protecting the particular zoospore when it is released into the external medium or water. Okay, then when in contact with the water, the vesicle quickly dissolves and you can see the mature zoospore released into the water medium. And this particular zoospore is having two portions. One area at the tip, you can see it is a pear-shaped structure. This zoospore is a pear-shaped structure, and it is having a particular tip, okay, an anterior tip, and this tip is hyaline, or it is clear and translucent in nature. So this particular anterior tip or hyaline tip, its area will be surrounded by, okay, just above the tip, that area will be surrounded by basal granules from which flagellus arises. Okay, so this particular mature zoospore will be having two parts, the upper part and a lower part. The lower part is called a hyaline tip or a translucent region. This translucent tip will go and attach with a substratum, a suitable substratum that may be on the top of another plant when it is an epiphytic species, or in the rocky bottom of that particular freshwater aquatic ecosystem. So in either case it will be attached with the substratum, and when attached with the substratum with the hyaline tip, this particular tip will form a holdfast or a basal cell. You can see this basal cell which is non-green. Okay, it is not having any chloroplast or something like that. So it is forming, that particular tip is converted to a holdfast, and the upper region, okay, that region above the hyaline region, that will be dividing to form a new filament, and it will form like this. Okay. This is all about the asexual reproduction in Oedogonium. In the next lecture, we will see about the particular sexual reproduction in Oedogonium. See, I have attached a brief note also here so that you can make short notes about Oedogonium asexual reproduction. Fine. Thank you.